Hello, everybody. Welcome to Ms. McGuire video lectures. And today we're covering the chemical level of organization part 2. So make sure that you watch part 1 first, right? The video became too long and I decided to divided it into 2 parts. So we're gonna skip the beginning of the PowerPoint and we go straight to where we left in our first video. And I'm using OpenStack's Anatomy and Physiology book. So let me see. So we cover most of the chapter. So, okay, I think right there. Okay, let's go ahead and begin. So we discussed in the previous video what organic molecules are. Well, really briefly, organic molecules are made of carbon and hydrogen. And on this slide, we will look at the chemistry of carbon. Carbon has 4 valence electrons, and it really makes it like unique atom. So you need to you're short of 4 electrons, so you have equal chances. So you want to donate them or you want to accept them. So actually carbon can form 4 covalent bonds with another atoms, and carbon has this ability to form very long chains, and those chains can be linear or they can branch, and this allows carbon to form really huge organic molecules. But the simplest organic molecules are hydrocarbons. So it's pretty much only made from carbon and hydrogen, like this. Like carbon form 4 covalent bonds with hydrogen. This is the simplest organic molecule, methane. That's a hydrocarbon. And then instead of one of this hydrogen or 2 or 3 or 4, you can add different functional groups. And this will make the difference in the type of organic molecules that you have. Now, what functional groups we have? We have hydroxyl group, that is OH. This is a polar group, and they are component of all 4 types of organic compounds that we will discuss in a minute. It can be carboxyl group, and this group found within fatty acid, amino acid, many other acids, right? Amino group found in amino acids, that is building block of proteins, methyl, phosphate, right? So the point is the carbon forms covalent bonds with hydrogen. If you replace hydrogen with other groups or with another carbon, you have a different organic molecule, and they can be really huge. And then we call them macromolecules. So let's look at the 4 major groups of macromolecules that we're gonna discuss today. And the first is carbohydrates. Carbohydrates. So they made of carbon, hydrogen, and oxygen, and it's always in the same ratio as shown here, like carbon and water molecule, H2O. And then you can take it as many times as you want, right? N number of times. So you will have an equal number of carbon and oxygen, and you will have twice as many hydrogen ions in a carbohydrate molecule. So here we're gonna discuss 3 forms of carbohydrates: monosaccharides, disaccharides, and polysaccharides. And you can see example over here of monosaccharides, glucose, fructose, and galactose, that are hexoses. And hexoses means they are made of 6 carbon atoms. So it's actually like a chemical formula for all of them is C6H12O6, right? But the structure is different. And that's a structure that gives those molecules slightly different chemical properties, right? So you can see we have same number of carbon and oxygen and twice number of hydrogen. We also have pentoses, and pentoses means they are made of 5 carbon atoms, and 2 very well-known are ribose and deoxyribose, and they are part of our DNA and RNA. Next group of carbohydrates is disaccharides. So the pretty much, if you have 2 monosaccharides and they are bonded together by these glycosidic bonds, so those are covalent bonds, then you have now disaccharides. So you have 2 simple sugar together, right? So if you have glucose and fructose bond, then they will form sucrose, and that's your table sugar. If you have glucose and galactose bond, then we have lactose, and you would find it in milk, right? And if you're lactose intolerant, you cannot buy regular milk. You need to buy lactose-free milk, because people who are lactose intolerant, they are missing this enzyme that can break chemical bond between glucose and galactose. So pretty much you drink milk, but you cannot absorb it into your bloodstream, because you can only absorb simple sugar in your blood. You cannot absorb from your digestive tract to your blood vessels, to your blood, disaccharide. And if you cannot digest, it's gonna just move through your GI tract, and it can irritate it. It can cause gas and pain, right? So that's what lactose intolerance is. If you take 2 glucose molecules, bind them together, you would form maltose, right? And this is sugar found in beer, right? Okay, so anyway, to consume in your diet, you need to split disaccharides into monosaccharides, and you have enzymes, luckily, to do it for you. Then polysaccharides. Polysaccharides include molecules that are made from many simple sugar, right? And it can be like just a single kind of long molecule, or it can branch out, or can form like a fiber looking structure, right? But those are all glucose binded together to another glucose molecules. So starches are plant-based food, easily digestible, glycogen stored in animal tissue. So plants store sugar in the form of starch. You and me store sugar in the form of glycogen. Plants also make cellulose, and this is what we call fiber. So if they tell you need fiber in your diet, that means you need cellulose. And it's interesting, we cannot digest cellulose, but we still need it in our diet to pretty much kind of like exercise your intestine. Like intestine, in the wall of your intestine, you have smooth muscles. And as any muscles need to, contract. You need to exercise muscles to keep them healthy. So cellulose, even you do not digest it because you don't have digestive enzymes to do it for you, it's still part of a healthy diet, right? Carbohydrates are a source of energy for people, right? So you have starch, you can consume, and then you use the starch, so you digest it into glucose and you store it in the form of glycogen, and you can use it for ATP production to power your muscles, to power your neurons, right? And this process of making ATP from sugar is called cellular respiration. And here the the equation for cellular respiration when glucose is broken into carbon dioxide, water, but those are kind of byproducts. The main product of cellular respiration is ATP. Cell structure, mostly for plant cells because they have a cell wall made of cellulose, but we also require carbohydrate because we require sugar because we have glycoproteins, proteoglycans, we have this glycocalyx, and glycocalyx is sugar covering of your cells. So you do need sugar to build your cells as well. Another group of macromolecules, organic macromolecules, are lipids. Now, I forgot to mention that carbohydrates, they are polymers. Polymers means they made from repeating units that we call monomers. So you have monomer, 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 you put them together, you have polymer. Lipids, they are not polymers, they're just macromolecules. Now, lipids are one example of lipids is triglyceride. Triglycerides are examples. So we do have other form of lipids like steroids, for example. They also lipids, but they have completely different structure. Triglycerides, however, they are a source of fuel. So that's a source of your energy. And they're made of glycerol, that is organic alcohol, and fatty acids that are attached to glycerol, right? And if you have 3 fatty acid chains attached, then we call it triglyceride or neutral fat. And this is done through dehydration synthesis reaction. Now, it's interesting that you burn triglycerides while sleeping. Yeah, so good 8 hours sleep is, just so important for your body. And you even can lose fat when you sleep, right? Well, if you think you're gonna lose like 10 pounds, that's probably not gonna happen, but do this experiment. Weigh yourself before you go to bed and in the morning, and you're gonna be lighter in the morning. So yeah, so sleep is, pretty good at burning some fat. And you will burn triglycerides during prolonged and slow activities, right? So actually, if you exercise, if you want to lose a couple pounds and you exercise only for 20 minutes, you're not going to burn fat, or unlikely. It's unlikely that you will burn fat. You will use your carbohydrates for energy. But if you exercises either very, very intensively or for long time, and you don't need to have like very strong exercises. It can be slow activities, but just for a long period of time. Then when you start burning fat. Okay, also lipids needed for vitamin absorption. We have lipid-soluble vitamins and for cushioning of your body. So a little bit fat is necessary. We do have 2 types of fatty acids. We have saturated and unsaturated fatty acid. And the difference is, now in a saturated fatty acid, all carbon atoms are form bonds, chemical bonds with maximum amount of hydrogen. So saturated means they're saturated with hydrogen. You have as many hydrogen atoms as you can, right? So if you look here on a top picture, each carbon molecule has like 2 hydrogen and this last one has 3, right? Now, unsaturated fatty acid would be when instead of hydrogen, carbon atoms form double bond. Carbon atom forms double bond with another carbon, as shown over here. And that's why you have less hydrogen over here, right? And another thing that happens, you have this kink in the molecule, right? It's not straight. Like, look, this molecule is straight. This one has like a little kink. And unsaturated fatty acids are healthier compared to saturated. And actually, your body does not require any saturated fatty acids. If you only eat unsaturated, that would be sufficient. You will synthesize all the vitamins and all the hormones that you need from unsaturated fatty acids, right? So what would be example? Well, unsaturated fatty acids, they are liquid at room temperature. So your oil, that would be example. Saturated, they are solid at room temperature. So that would be a butter, right? What are trans fat? Everybody heard about trans fat, and it's really bad. And trans fat, it's interesting, it's actually unsaturated. So look, this is a trans fat shown right here. So you have this double bond, and you would think like, oh my goodness, that's good, right? Because I want this double bond. Like you have double bond over here. But see the difference. This is hydrogen ions on the same side of a bond. And here, hydrogens are on the opposite side. And that's what trans means. So hydrogen is attached across the double bond. And even this molecule is unsaturated, it's still straight. It doesn't have this kink like nice this one has. This is called cis fat. So what is the point of being unsaturated if you still like straight molecules? So this can clot your blood vessels. This is if these molecules affect your cholesterol level, right? Or low density cholesterol. So this is, and especially trans fat, well, trans fat for some reason is especially bad for our diet, right? So example of trans fat would be margarine. Yeah, so just eat natural butter if you want something solid. Also, processed food contains a lot of trans fat. So anyway, we have saturated and unsaturated fatty acids. Another example of lipids would be phospholipids, steroids, prostaglandins. All right, so so here's the example of phospholipid. You still have this glycerol, right? And instead of 3 fatty acids, you have 2, and 1 acid is replaced by a phosphate group. And because of this, the phospholipid molecule have very interesting characteristic. They bipolar molecules, so they have a polar head and non-polar tail. And this chemical property is very important in formation of bilayers of phospholipids. And you know, cell membrane is mostly made of phospholipids. So because it's a polar head, it will be attracted to water. And because tails are non-polar, they will repel water. And this makes the phospholipid kind of like, look, if you have phospholipid and the tails, and you have another phospholipid, right, with its tails. So the tails will face each other and the heads will face where the water is and where we have water. This will be extracellular fluid, right? And this will be your cell, right? So intracellular fluid, right? So here how you form a cell. So here's a cell, right? So here's the nucleus. So you will have fluid inside and fluid outside, and phospholipid molecules will organize themselves in a pattern when, you know, the tails are facing away from water, right, and heads are facing towards some solution, watery solution. And this would allow us to form double layer, right? Nice double layer, and that's the formation of a cell membrane. If you look at steroids do not look anything like phospholipids or triglycerides, right? All steroids, they have these 4 rings made of, carbon chains and then some extra parts on it. But steroids share lots of properties with another lipids, right? Like they water insoluble, they can pass through the cell membrane. So that's why they are in the lipid group. An example of steroids would be cholesterol, and cholesterol is the basic for all steroids formed in our body. But what are those other steroids? Like important hormones, testosterone, estrogen, progesterone, vitamin D, right? So they all are formed from cholesterol. And cholesterol is also needed to build the cell membrane around your cells. So it's a part of cell membrane. Another example are prostaglandins. Now, prostaglandins are molecules that are released when your cells are damaged. And they like inflammatory chemicals. Let your body know that there is some damage of the cell, there is some inflammation. Okay. And proteins. Well, proteins are amazing molecules. Most of your body is made of proteins. If you take like, okay, carbohydrates, lipids, proteins, nucleic acid, most of your body is made of proteins, right? So proteins are also polymers and monomers. So those building blocks of proteins are amino acids. And they join together by covalent bonds that we call peptide bonds. So here's the basic structure of an amino acid, right? When we have alpha carbon or central carbon, we have amino group and carboxyl group. And what is different is this R group or side chain group. So whatever group you put here, you will have a different amino acids. Now, when amino acids are connected through those peptide bonds, they form what we call primary structure of protein. It's just a chain of amino acids. Sequence of amino acids is extremely important. If you have error in the sequence of primary structure, you might have like some genetic disease or condition like sickle cell anemia, for example, right? But a primary structure of protein, it makes protein, but it's not functioning protein. Protein need to be folded precisely into secondary and tertiary structure, and sometimes even in quaternary structure. So the secondary structure can be either alpha helix or pleated sheet. And here we have this hydrogen bonds, right, between different amino acids, between different parts of the those monomers, right? So those monomers start acting with each other, forming hydrogen bonds, and it's either start looking like a helix or like a pleated sheet is like if you want to make a little fan from a piece of paper, that's how it would look like. And then when you have alpha helices and pleated sheets together, then it's called tertiary structure. And this is polypeptide, right, with its tertiary structure. It can be functional protein at this point, but some proteins, they are made of several polypeptides. So we have polypeptide with a tertiary structure, and then they come together, several of them, and then we have quaternary structure. So for example, hemoglobin, it's a globular protein, functional proteins that made of 4 parts, 4 polypeptides. Not every protein has tertiary structure, or not every protein has quaternary structure, but all functional proteins have tertiary structure. And what is the nature let's say if you heat proteins or if you change pH of these solutions, and proteins start losing this structure. That's what we call denature, and they stop functioning. And sometimes this denature of protein is irreversible. Sometimes it can, protein can fold itself back, but in many cases they can't. And that's why high fever is so dangerous. And they tell you if you have 103, right? For your kids, or 104, you need to go to emergency room because your temperature goes up and proteins, especially enzymes in your body, start losing their structure, and they losing structure cannot function, right? And if it's reversible, you can die. So changes in the temperature or pH can drastically affect shape of proteins. And at some point, when we learn how to reverse this denature, then I don't know, it's a lot of stuff might happen, because the death, when we dying, right, our proteins denature. Now think if you can reverse them back to normal proteins, maybe we can start functioning again. Who knows, right? Okay, so what are examples of proteins in your body? Enzymes. Enzymes are proteins. So what enzymes do, they speed up chemical reactions. And whatever reactants are, we call them substrates. So substrates will bind to enzyme, to active site of enzyme. It's called induced feed. That means when substrate binds, the shape of enzyme changes. And enzyme will catalyze this chemical reaction between two reactants, right? So here you have your product. Product is released, enzyme is not consumed in this reaction. So it's ready for new substrate, right? However, we do have enzyme inhibitors. We call them poisons. Enzyme inhibitors are chemicals that prevent a substrate from attaching to an enzyme. So if you look here, you see this enzyme shown kind of in blue color. Anyway, whatever color is that. And enzymes, they have active site. Active site means substrate will bind here. Any other site where other molecules can bind, called allosteric site. Now, what if we have a molecule in your body that's gonna bind to this active site and block enzyme from binding to substrate? Then enzyme cannot catalyze reaction, right? That's enzyme inhibitor. And it can be either competitive inhibition, when inhibitor binds to active site, or non-competitive inhibition, when inhibitor binds to allosteric site, but that will cause active site to change its shape. So substrate still cannot bind. And that means no chemical reaction. What would be example? Cyanide. Well, cyanide is really dangerous poison, right? And what it does, it interferes with enzymes that needed in cellular respiration, cytochrome excitase. And cellular respiration needed for ATP production. No ATP production for couple minutes, 5-8 minutes, and your brain start dying. That's why cyanide is so dangerous. When I first heard about it, I was thinking cyanide, carbon and nitrogen. Our body is made of carbon and nitrogen. How it can be dangerous? What possibly this cyanide can do to us? It's a carbon and nitrogen. But yes, that's gonna inhibit enzyme, and that will prevent your cells from making ATP. If your cells don't make ATP, they start dying very, very quickly. And first cells to die will be neurons. Also, we have inhibitors that inhibit enzyme needed for your muscle contraction, right? And then it can cause muscle paralysis. However, we can use those enzyme inhibitors in the medicine. If we don't want some reaction to happen, we can prevent them by using those poisons. For example, antibiotics. Why we can use antibiotics? Because they do not affect our enzymes. They inhibit bacterial enzymes. And bacteria cannot produce structures called peptidoglycan. And without peptidoglycan, bacteria cannot make a cell wall. So bacteria is dying if you're using antibiotics because bacterial enzymes are inhibited, right? Aspirin and Advil, they inhibit enzymes that convert phospholipid into prostaglandins. We just mentioned prostaglandins. Those are inflammatory chemicals. So if you block the enzymes that that help you to make this prostaglandin, you decrease the number of prostaglandins, you decrease inflammation. That's what aspirin and Advil do. Lipitor. Like my mom is prescribed Lipitor. What Lipitor does, it inhibits enzymes that convert fatty acid into cholesterol. So because my mom has high blood pressure and her cholesterol level is high, now they're giving her poison that inhibits her enzyme and doesn't allow her cells to convert fatty stuff that she eats into cholesterol, such decreasing level of cholesterol in her blood and decreasing her blood pressure, preventing hopefully from, strokes and heart attacks, right? So enzyme inhibitor can be used in medicine. Another example of proteins are hormones and antibodies. Most protein hormones must be injected. Otherwise, you're going to digest them. Steroid hormones are not broken by your digestive system, but protein hormones, you just digest it like a regular steak. Example of protein hormones would be insulin or growth hormones. In antibodies are proteins produced by B lymphocytes. Not precisely B lymphocytes. B lymphocytes become plasma cells, and plasma cells produce antibodies, and antibodies inactivate your bacteria or other pathogen in your body. And that's it for proteins. Next group of macromolecules are nucleotides. Nucleotides are actually your DNA, your RNA, right? You heard about it. So nucleotide, your DNA and RNA are not nucleotides. They are made of nucleotides, right? They are nucleic acids. So nucleic acids are polymers made of individual nucleotides, right? That makes sense? So nucleotides are building blocks for your DNA and RNA. And we have 4 nucleotides, right? We have adenine, guanine, actually 5, sorry, adenine, guanine, cytosine, and thymine. In RNA, you have uracil instead of thymine. So in DNA molecule, you have 4 different nucleotides. In RNA molecule, you have 4 different nucleotides, right? But we have total 5. That makes sense, right? Because RNA has uracil and DNA has thymine. And each nucleotide is made of pentose sugar, ribose or deoxyribose, phosphate group, and nitrogenous base. So that nitrogenous base, that is different. So sugar is the same, phosphate group is the same, and nitrogenous base is different. And this is what make these 5 types of nucleotides. 5 types of nucleotides. Okay, so nucleic acids, polymers of nucleotides, we have DNA and RNA. And very briefly, without going into details, DNA is double helix, and DNA stores information about protein synthesis, and RNA is single-stranded molecule, and it's also involved in protein synthesis. Right, so it's helping your ribosomes to make proteins, right? So they both are involved in protein synthesis. DNA is a large molecule found in the nucleus of your cells, and RNA is smaller molecule. We have different types of RNA, like transfer RNA, like ribosomal RNA, messenger RNA, right? But nevertheless, both molecules are involved in protein synthesis. That is extremely important for normal functioning of cells. And ATP. ATP is a nucleotide, right? It has sugar, ribose. It has a nitrogenous base, specifically it's adenine. That's why we call this adenosine. Kind of adenine and ribose together give us word adenosine. And then depends how many phosphate groups are attached. If we have 3 phosphate groups, then it's ATP, adenosine triphosphate. Right, but we can have 2 groups attached, then it's going to be ADP, adenosine diphosphate, or only 1 phosphate group attached, then it will be AMP, adenosine monophosphate. And importance is, well, ATP is energy currency, right? It means your cells directly use ATP. Like, what does it mean directly? Well, if I eat steak or I eat pancakes, I use this food for my energy, but I cannot directly use them, right? I cannot directly use even simple sugar or amino acid to power my muscles. I need ATP to do it. And the energy that you use is stored inside these chemical bonds between phosphate groups. And the last one, this kind of like the second bond, is the richest in energy. So usually ATP is broken into ADP, like over here, you see ATP hydrolyzed into ADP and inorganic phosphate. And that's energy that was stored in this bond, used to contract your muscles, to send electrical signals, right? For your eyes to work, for your retina, you know, to be able to process this, have this, you know, phototransduction, transduction. Yeah, that's a funny word. Anyway, so you need ATP for many processes happening in your body, right? For movement, for digestion, for filtration, for secretion, you name it, right? And then through the cellular respiration, this phosphate group is reattached back. That's called phosphorylation, and you make new ATP and you store energy. So really, I would compare ATP with a battery. So if you have a battery, but it's a chargeable battery, so you use it, then you charge it. You use it, you charge it. You use it, you charge it, and this way you have this energy currency available to power your cellular work. Okay, and I believe, oh no, this is the last one. Okay, one more slide. And this slide really shows us how ATP is used, right? So for example, for transport work, right? So if you have a pump, if you need to move stuff against its concentration gradient from low concentration to high concentration, right? Over here, ATP attaches to this protein, phosphate group is removed, this protein changes its shape and molecules move from one side to another side, like let's say across cell membrane, right? Or smooth muscles relaxed, you have hydrolysis of ATP, energy is used for contraction or chemical work, right? And phosphorylation is when terminal phosphates are enzymatically transferred to and energize other molecules. That's phosphorylation. Oh, okay, so, you know, phosphate are transferred and energize other molecules. Such primed molecules perform cellular work using the phosphate bond energy. Okay, so anyway, I hope it all makes sense. So why are we discussing ATP over here? Because ATP is nucleotide, right? And it's kind of like our last group of organic molecules, nucleotides and nucleic acids. Only nucleic acids are polymers of nucleotides. ATP is individual nucleotide with phosphate groups. And those phosphate groups store lots of energy in the chemical bonds between them. And when phosphate is transferred between ATP to maybe other molecules, right? When we have this hydrolysis of ATP into ADP, we can do transport work, mechanical work, or chemical work, right? And if you're confused about phosphorylation, then maybe I should record another video, maybe short one, to explain how it really works. Or you can find another video on YouTube, right? Because here it's just like really brief definition, right? So when we transfer terminal phosphate to another molecules, we kind of energize those molecules and they can change their shape, right? Either, like this protein, or maybe pull, like actin gonna pull myosin towards the middle of sarcomere. So it's very interesting, but a long and complicated topic. Okay, so I think that was our last slide over here. So remember, this is part 2 of chapter 2. Don't forget to watch part 1. And oops, I'm moving different stuff over here. I don't know why. Thank you for watching, and I hope it was helpful.